high-risk obstetrician at Mass General Hospital, and we're excited to share with you the findings of our new study being published in JAMA Network Open. The main findings of the study were that women who have a SARS-CoV-2 infection, the virus that causes COVID-19 in the third trimester of pregnancy, have very low rates of having the virus in their bloodstream. We found that out of 64 of these women, none of their placentas were infected with SARS-CoV-2. We found no neonates who were infected at the time of birth, which can be potentially reassuring information to women. And then we found a very interesting and unexpected finding that the passage of the mom's antibodies, or the proteins that fight the virus, to the neonate through the placenta and the umbilical cord was less than expected. And it was less than what we saw for antibodies to influenza, which women routinely receive vaccines to in pregnancy, as well as against pertussis, again, which women routinely are vaccinated against in pregnancy. So these findings were, on the one hand, reassuring, but on the other hand, maybe concerning in that babies may not receive as much protective antibody from the mom against COVID-19 as they do against, say, the flu. We did this with a large research team that spanned several institutions in Boston, although it was based here at Mass General Hospital, and also spanned a wide range of expertise, including placental pathology and including immunologists and virologists. So several of those people are joining us here today and will kind of describe different aspects of the study. So first going to Dr. Roberts, who's a placental pathologist here at MGH. Dr. Roberts, would you share with us more about what you learned from these placentas?
Sure. So we looked at probably the largest number of placentas of SARS-CoV-2 infected women or COVID-19 complicated pregnancies. And what we looked for were evidence of placental infection and also damage to the placenta from mom's infection. And what we saw was that none of the placentas we looked at had evidence of placental infection, which is very reassuring, as you said. But we did see a definite trend towards damage in the placenta, what we call maternal vascular malperfusion, which is damage from the mom's side of perfusing the placenta.
In addition, Dr. Roberts' team discovered that the two different receptors, or protease and receptor, that are necessary for the placenta to get infected are both physically distant in the placenta, and their patterns of expression don't favor placental infection. And then Dr. Alter's team discovered that the passage of antibodies across the placenta from the mother to the neonate that occurs in COVID-19, those COVID-19 specific antibodies are at a level less than what we expect to see.
What the placenta does is it captures the best and most functional antibodies from the mom and transfers it over to the baby to make sure that the baby, on the day that it is born, it is empowered with the best possible immune defenses against pathogens. So of course, when SARS-CoV-2 began to emerge in pregnant populations, the first thing we did is ask ourselves, are the antibodies getting transferred? Because that would be, a really important sign and reassurance for those, neonates that are entering the world almost completely vulnerable if they don't have those antibodies. And typically for influenza and for pertussis and other pathogens, as you mentioned before, that we vaccinate against to really push those antibodies up to higher levels to really empower our neonates. There we would see that, mums and antibodies are here, and the baby's antibodies were up here. So you can see the placenta really dragging those antibodies out and giving them over to the baby. Now, that was not the picture we saw with SARS-CoV-2, and that's what I think got us a little bit worried. Because in SARS-CoV-2, the mom's antibodies would be here, and then the baby's antibodies were down here, or they would creep somewhere below where the mom's antibodies was. And so what this is telling us is that the placenta may not be able to capture these antibodies as well as they do for the flu antibodies or the pertussis antibodies. The good news is that because the flu and pertussis antibodies were going over, it meant the placenta was relatively intact. It was functioning. But what it told us is that there was something funky about the SARS-CoV-2 antibodies that we had to really begin to pay attention to, so we can make sure that as we move forward in the pandemic, that our vaccines can do better than natural immunity to protect those neonates.
Thank you. Yeah, that was really surprising to us. And, I remember speaking with you about that finding in the late spring, early summer, and repeated it several times to just make sure that that was true because it was so unexpected. And then when we found that was true, we really wanted to let people know about that because that is a risk to those neonates. And then Dr. Anjali Kaimal is with us, and she's another of the last authors on the paper and is the director of clinical research for obstetrics and gynecology at Mass General Hospital, and has a leadership role in all obstetric research that takes place there. So I was wondering if you could tell us, what do you think are some of the implications of this for how we counsel women or about women who are pregnant for SARS-CoV-2 in general?
I think the first important thing is that this is another great reminder of, first of all, how miraculous pregnancy is and how it both, there are changes that protect us as we're pregnant, there are things that protect the baby, but also to remember that with each new pathogen we encounter, we really need to be studying pregnant women to understand how that complex physiology and pathophysiology works. We were so lucky to have people like Dr. Alter and Dr. Roberts to collaborate with, who have great expertise in looking both at the placenta and in immunology, and our other virology colleagues as well. But what we realize is that each new virus that we encounter acts a little bit of a different way. So I think that remembering that pregnant women are such a priority, critical population to be studied whenever any of these challenges is ahead of us is really important, and something that this reinforces. I think after looking at that, the first thing that we've talked about already is that women can be somewhat reassured by this. So we know that the likelihood of vertical transmission is low, not zero, but we here can demonstrate some of the mechanisms behind that, which also may help us to understand which are women who might be at risk for those rare complications, including vertical transmission. And importantly, what are the things we can do to try and reduce those risks? And then, as Dr. Alter mentioned, all of this is so important now as we're looking at the vaccine, because we know that there are ways in which vaccines may be more immunogenic than native infection. And what we want to make sure is that the way that we vaccinate pregnant women ensures that they are protected from the severe disease that they're at risk for, but also that we're able to give that extra boost to the neonate, particularly so that they're able to carry it into that important time in the first part of life when they're otherwise unprotected.
So it's really been a group effort to learn these new things about COVID-19 infection and pregnancy. And really what we found were that pregnant women had very low rates of transmission to the neonate. We actually found 0 cases, but we know that the rare case does exist. We found some hints as to why that might be. First of all, because pregnant women rarely get a bloodstream infection with COVID-19. So even if we can detect the virus in their nasal fluids or their saliva, we don't see it in the blood. And because it's not in the blood, it's less likely to seed the placenta. In addition, Dr. Roberts' team discovered that the 2 different receptors, or a protease and receptor, that are necessary for the placenta to get infected are both physically distant in the placenta, and their patterns of expression don't favor placental infection. I think a special thing about Mass General Hospital is just the wide availability of expertise across a broad range of specialties, and the kind of marriage of clinical research with basic or bench science, and the insights that we can get about disease that are unique in this way and that span disciplines. So I've been really grateful to be part of this amazing team with Dr. Alter, Dr. Roberts, Dr. Kaimal, Dr. Jonathan Lee at Brigham and Women's Hospital, Dr. Katie Gray, and Dr. Iris Collier at Beth Israel Deaconess.